Good afternoon, everyone. Thank you for joining this exciting Environmental Health Sciences seminar. I'm Ellen Hahn. I direct UK Cares, and just a quick plug for our recent request for applications that just went out this week. We do have pilot funding, so yay, please apply. We have high impact and early stage career awards, and so please, if you don't, I'm sure Carol will put that link in the chat, and also a plug for our research portal. So if you need assistance with samples or methods or whatever, just a consultation, please fill out a request on our portal. So today's talk is, I can't wait. I'm going to turn it over to Kevin Pearson, who's our career development lead, and he will introduce Todd. Thanks, Kevin.
All right, thanks, Ellen. And I just want to quickly introduce Dr. Todd Everson from Emory University. And Todd, just to let you know, we all do keep our cameras off just to kind of keep the signal great. So we are paying attention. Todd does have to give a lecture after this, so he's going to keep his talk in the 30 to 35 minute range, so we have some time for questions and discussions. Todd is a PhD and also has his master's in public health. He's in the Department of Environmental Health and Department of Epidemiology at Rollins School of Public Health at Emory University. He earned his bachelor's in environmental health at Colorado State University in Fort Collins, Colorado in 2006, then went on and got that master's in public health at in health disparities at Oregon Health and Science University in Portland, Oregon. Then he got his PhD in epidemiology at the University of South Carolina in Columbia, South Carolina. Then he went on and did a postdoc with Dr. Carmen Marcet, who's a close friend of mine. And he was at Dartmouth with Carmen and then moved down to Emory with Carmen. And then he gained his faculty position there in 2018. So Todd has been very productive. He has is multi-PI on 2 different R01s, including the Victor grant that I'm MPI of as well. And I will say that without Todd, we never would have gotten that grant funded. He actually pulled an all-nighter and rewrote all of the bad grantsmanship things that I had in the grant and redid it. And we got the best score out of that entire study section. So if it weren't for Todd, we wouldn't have that grant, which I think he'll probably mention at some point today. And I also wanted to mention another highlight about Todd is that he's in the first percentile club. So Emory has a first percentile club for those individuals that receive a 1 score on an R01 at Emory. So Carmen tells me that he's not even a member of that club yet. So he's hoping that he can achieve the same things that Todd has done. Todd is a prolific publisher. He's already got close to 50 manuscripts. And we did bring him to UK in 2017 to give a talk. And Todd and I were able to really get closer during that visit. So just for any of the trainees out there, make sure you start to build up that network because you never know when it's going to pay you back. And since Todd doesn't have much time, I'm going to go ahead and let him take over. So Todd, take it away. Thanks.
All right. Thanks, Kevin. Thanks for that great introduction. So we didn't have to rewrite all of your sections. Some of them were pretty good. The title of my talk today is The Early Life Environment, Epigenomics, and Children's Health. And I'm kind of going to focus more on I'm going to give an intro here. And then that intro is going to start with what's the framework that I view my research through. I'm going to do a brief review of DNA methylation because I think a lot of people have at least some familiarity with that now, but I want to be sure we're all on the same page for some definitions and foundational ideas. Then I'm going to talk about my work with placental epigenomics, why I'm kind of focusing in on some of this area. I'm going to highlight how we've been studying it with tobacco exposure. And then at the end, I'm going to have a few highlights related to our Victor project and the ongoing and future directions with my group. And so for my background and kind of my framework, my trainings first was in environmental health, then healthcare disparities, epidemiology, and then postdoctoral training in epigenomics. And when I have it all listed out here, there's a lot of E's and EH's, and it all looks like some kind of like plan of things that really fit well together. But I will say that as I was going through it, it was kind of an adaptive zigzagging training experience where I was able to adapt and make use of opportunities that were presented to me. And the reason I highlight that is, my first training in environmental health was during my bachelor's degree. And by the time I was still studying EH during my postdoctoral training, where I was really focused on epigenetics, the field had actually changed quite a bit. So when I was getting my training as a bachelor's student, that was really where EH was still kind of the OG EH, where everyone was really focused on chemical contaminants in the environment and how those can affect health. But the current view of EH is much more expansive and broad, and it includes a lot of other factors that can impact health that are kind of unrelated to genetics. And that can include psychosocial factors, built environment. It still definitely has a big focus on chemical contaminants. But as this field has grown and expanded, it's kind of now moving in this direction of this exposome idea, where it's not just about single exposures or individual exposures at a particular developmental window, but exposures across the life course. And not only about the exposures themselves, but how are those internalized? How do those have biological responses? And then are those biological responses important to health? And so this idea of the exposome was kind of growing as I was doing my postdoctoral training. And that's really where I solidified my interest in epigenetics, environmental exposures, and children's health. And the reason that epigenetics became a lot of my focus was that I was doing a lot of reading and work in the developmental origins of health and disease area. And that's basically saying that we have these early exposures that can have a long-term effect and lead to adult disease. Now, if that's the case, once that exposure has been removed, there has to be some internal biological response that is either a change in function or damage that then has persistent effects across the life course. And epigenetics is one of the mechanisms that could help us understand that process. And so to give a very brief overview of epigenetics, this field really refers to any of the chemical modifications to the DNA that don't alter the sequence itself, but they kind of change the way that the genome is being brought to life, because the DNA is a static molecule by itself, but with the different chemical modifications to it, it can kind of become much more dynamic. And the way to think about this is we all have sort of one DNA sequence for the most part within our different tissues, but then within our different organs, you see very different structures being formed, very different biological processes being activated or repressed. And a lot of that is controlled through these epigenetic mechanisms. I'm going to primarily talk about DNA methylation, but there are a number of different ways that these epigenetic features can control gene expression. And so I'm highlighting three of the most common ones here. DNA methylation is where you have these methyl groups being added to single nucleotides on the DNA itself. And this can allow for fairly local regulation of genetic activity. You can also have histone modifications, and this is histones are these molecules that bind together to form something called a nucleosome, which looks like these sort of blue hockey pucks here. And the DNA is wrapped around these nucleosomes so that the DNA can be organized and compacted and stored within the nucleus of a cell. If it was all just kind of waving around in its long form the entire time, it would just take up a ton of space and it wouldn't be organized in a way that you could kind of have targeted identification of specific genes for expression. And so these histones allow for organization, but then the chemical modifications to the histone can cause the DNA to unwind in specific locations that allow for the cellular machinery to interact with it and then have that sort of localized specific gene expression. And then lastly, chromatin organization is more the broad way that the DNA is organized when it's in this sort of wrapped around nucleosomes, this structure. It provides the bigger picture of that structure. And so chromatin organization can have a bigger effect on wide regions of the genome being accessible or inaccessible to cellular machinery. Now, most epidemiologic studies focus on DNA methylation for a couple of reasons. This refers to the chemical addition of a methyl group that predominantly occurs at cytosine bases when they're followed by a guanine. And so we call that a CpG site. So you'll hear me use that terminology a lot. And that's just talking about these specific locations on the DNA. Here you can see the purple and the orange. This is CpG sites. And it predominantly occurs at the C. And at these CpG sites, that C can either be methylated or unmethylated. And these methylation patterns are typically retained on both strands of the DNA. And what that means is that during replication, the DNA will get split. You have two new cells formed. You have that new strand of DNA writes the complementary sequence. And during that, it's able to maintain the methylation pattern on the opposite strand. And so one of the biological implications of that is that if you have an epigenetic pattern established early on while cells are really proliferating and dividing, there's a chance for that sort of methylation pattern to get encoded and carried forward as that organ grows and develops. And the last thing I want to mention about methylation itself is that this is a normal biological process. People that are kind of outside of this field will often see papers talking about, smoking or a particular diet or a behavior are associated with DNA methylation. And so if you're not reading the papers more closely, it kind of gets this impression that methylation is a bad thing. It happens with exposure. That's not the case. It is a normal biological process. And so we're not saying that the accumulation of methylation is necessarily bad or good, but if it's occurring in a way where it's aberrant to the way it should be occurring. So if there's a pattern where a gene should be expressed and the methylation pattern allows for that, if you identify a group of people where now the methylation pattern is different and it's repressing that gene from being expressed, that's where it can become a problem. And so it's not the presence or absence, it's more, is it occurring where it shouldn't be, or is it missing where it should be occurring? On a real brief note on how we measure it, the gold standard right now is actually building on a lot of the work from the Human Genome Project. And so that was a multi-billion-dollar endeavor to identify how do we measure the entire human genome. And they developed a lot of technology and data storage and data analysis techniques in order to achieve the first human genetic sequence. And the epigenetic measures that we have now build directly off of that. And so in order to measure DNA methylation, pretty much what the vast majority of researchers do now is you apply, so you get your DNA sample, you apply this chemical conversion called bisulfite conversion. And what that will do is it'll convert a cytosine to a uracil if that cytosine is not methylated. Whereas if the cytosine is methylated, it will not be converted. So then we would amplify that DNA and then align it to a sequence. And if we expect a cytosine, but we identify a thymine, then we know that that's a location where the methylation, or where that CpG was not methylated. If we expect a cytosine and we detect a cytosine, then we know that's a location where there was methylation. And so this allows us to use all of this technology that was developed to really grow almost an entirely new field of epigenomics and do it very rapidly. And so the way a lot of my research program is built is my training is in epidemiology. So I primarily work with either existing or ongoing cohort studies where they're collecting biosamples. We're collecting lots of either survey-based or biomarker-based exposure data. We're extracting the DNA. We're applying this bisulfite conversion. And then most of the work that I'm working on right now is using a microarray to then measure methylation at a large number of sites throughout the genome. So we would call it genome-wide, but it is missing a lot of actual locations still. And this is the chip that we focus on for now, which is the Illumina methylation epic array. This chip is only a couple of inches in length. And here you can get 8 different samples from different people on here and measure 850,000 different locations on the DNA. So with this tiny chip, we have a huge density of information that we're able to generate. That chip is then read by a sequencer, and the sequencer outputs a data matrix that looks a lot like this. And so to kind of just visualize what are we doing at the data analysis stage, this is the most common type of study that's performed, which is an epigenome-wide association study. And so we'll have something like 850,000 rows of CpG information. In this example, 10 subjects, cases or controls. And we're just trying to identify, are there CpGs where we see very different methylation patterns between these two groups? And we can apply this in either an exposure, unexposed group, or with continuous exposures and outcomes as well. Now, there's a second field that's really developed a lot, especially over the last 5 or 6 years, which is applying machine learning techniques that doesn't really search as much for individual CpGs, but tries to build algorithms that are predictive of health characteristics or prior exposures. And I'll briefly talk about that towards the end. This is a much more rapidly developing field over the last 5 or 6 years. And so my program really focuses on a couple of different areas. One of those is perinatal exposures, methylation and birth outcomes. I've done a lot of work with metals. I have some ongoing work with air pollution. And today I'm going to highlight some of what I've done with tobacco exposure, although we have some ongoing work looking at other substance use as well. And for these studies, I like to focus on biologically important tissues. That seems like an obvious statement to say, but actually, when you think of epidemiologic studies, you have to work with the tissues that are available. And so in some cases, those can be very biologically relevant. So in the study of birth outcomes, growth, and perinatal exposures, the placenta is obviously very important to think about. But when we have access to lots of other potential tissues to measure, there might be ways to leverage those data that still tells us about health and development in an interesting way, even if it's not telling us about a causal mechanism. And so one of my other focal points is identifying epigenetic markers of health and development. And for this, we're using easy to collect peripheral tissue samples and applying these machine learning and prediction modeling to hopefully identify infants that are at risk of neurobehavioral and developmental issues in prospective studies. And so my focus on the placenta is, as a very brief background, is because if you're not as familiar with it, there's three really important functions that it tends to serve during pregnancy. One, it offers protection by preventing maternal rejection. It also protects against infections, and it can filter out some environmental toxicants. It's a very metabolic organ. It also controls the transfer of oxygen and nutrients to the fetus, as well as controlling the waste removal from the developing fetus. And it produces lots of metabolic products. And then it's also a very active endocrine organ, producing lots of hormones and growth factors. And it's one of the major organs that guides fetal growth and development. And it also influences maternal physiology. So it's this really important organ. And from an epidemiologic perspective, it's actually really nice that you can actually sample this organ at birth, because at that point, it's served its function and it's typically a discarded tissue. So it's able to be sampled, but it also serves a very critical role in growth and development during pregnancy. Now, disruption of those functions can lead to a number of problems, including maternal health complications like preeclampsia or gestational diabetes. And it can also lead to adverse birth outcomes like preterm birth and fetal growth restriction. And both of these birth outcomes are risk factors for adult onset disease. It's thought that this is because you're having some disruption to fetal programming that's carried with those children as they grow and develop, and then they're at just increased risk for some of these outcomes as adults. And so this is kind of the area that we're focusing on for prenatal environmental exposures, placental omics. And I'm going to do a real brief highlight on some of the unique methylation patterns of the placenta. As some of you that may study genomics are aware, at fertilization, the zygote is formed, and that's where you have this new DNA sequence formed from both the maternal and paternal DNA. And at that point, you get this very rapid demethylation, where almost all of the methylation patterns of that sequence are removed. And after about 10 days, something called the blastocyst forms. This is where you get this sort of cell growth of two different compartments. One is this purple layer called the trophectoderm. The other is this inner cell mass. And that purple trophectoderm is what becomes the placenta. The inner cell mass develops into the fetus. And so what's interesting is that up until this point, you kind of have a singular methylation identity, but then it diverges after the forming of the blastocyst, where the placenta follows a much more hemimethylated state, where at birth you have a lot less overall methylation of the placental DNA compared to the somatic tissues of the fetus. And so the reason I highlight this is when we're trying to study health and development and environmental exposures, which tissue we study is going to tell us very different things. And this just kind of shows that these follow two very different epigenetic developmental patterns. And one of the reasons for that is this unique feature called partially methylated domains, which are detected in the placenta. These are very long regions of the DNA where you have extremely high methylation followed by very hemimethylated states. And so you get these kind of blocks where if you measured it here, the density of methylation over that DNA sequence, you kind of see this really heavily methylated state, and then just a very sheer drop where you get this pattern of much more like some high, some very low methylation. So on average, it's about 50%. Now, partially methylated domains exist in other tissues, but they're just much more prevalent in the placenta. And normally, if you think of methylation as primarily being this repressive effect, that's not the only function it serves, but that is the most common paradigm that is viewed through. You would think that maybe these hemimethylated regions are these genetically active regions. And what's kind of interesting is we actually see the exact opposite of that, where the PMD regions are predominantly characterized by having low methylation within the gene body, but then very high methylation in the promoter, which is the sort of paradigm for methylation as a repressor. And so these actually are very representative of genetically repressed regions. And in the placental epigenome, these cover about 40% and about 4,000 different genes. And so this is very different from other tissues. And if we actually look at our methylation pattern, how different does it look? So this is called a density plot. And on the x-axis here is the average methylation, or the density of CpGs with a methylation state close to 1, so almost fully methylated, versus close to 0, which is almost fully unmethylated. And in cord blood, what you see is you have the vast majority of CpGs either fully methylated or fully unmethylated, and then you have a much smaller region here of CpGs that are in this sort of hemi-methylated state. Now, CpGs that are in this region, they just have a wider range of possible methylation values they can take on, and they're much more dynamic. So there's more of these that are likely to be associated with exposures and outcomes compared to CpGs in these two peaks. So how does that look when we compare it to the placenta? It's actually very, very different, where this peak on the right, it still exists. You have a larger number of sort of heavily methylated CpGs than hemi-methylated, but you can see that distribution kind of bleed into that hemi-methylated state more. And so what that means is that if you looked at the overall genome and we're trying to study DNA methylation on average, you would have completely different findings if you studied cord blood versus the placenta. But the other thing that we think it means is that you have this statistical characteristic of there's just a lot more CpG sites within the placenta that are likely to be responsive to the environment or associated with environmental exposures and potentially related to outcomes because they have so much more variability in their methylation state. Okay, so that's my last kind of general data presentation. And I want to highlight how we've been studying the placental epigenome by focusing on one of our meta-analyses of maternal smoking during pregnancy. And this is an exposure where we've seen one of the strongest impacts on the methylome, almost no matter what tissue you study. And so as a brief overview, we performed this international study with 7 different cohorts where we were testing whether or not the placental methylome was different between those exposed to maternal smoking during pregnancy. We first looked at whether they reported any smoking or any exposure at all during pregnancy, and then compared a stratified analysis after only looking at those moms that reported smoking throughout the entirety of pregnancy. We then did some functional enrichment to understand what's the biological impact of this response. Are they related to birth outcomes? And is this signature unique to the placenta, or is it similar to that of cord blood? Now, the motivation for this project, on average, about 10% of pregnant mothers smoke during pregnancy, but there's huge variation here depending on what region of the US you're in, as well as if you're comparing the US to Europe. And so it's actually still a very prevalent exposure, but it is very geographically dependent. And tobacco smoking is known to cause adverse pregnancy outcomes. It has already been shown to have a profound effect on the newborn epigenome. And one of the questions we're asking is it possible that this epigenetic response is an intermediate between the exposure and the outcome? And so the vast majority of studies up until this project focused on cord blood methylation. But I kind of set the stage here to say if we're studying cord blood methylation, that's primarily composed of immune cells. So would you really detect the mediated effect on growth in immune cells? You might detect some of it, but I would think focusing on a growth regulating organ like the placenta would be a better target for identifying a true mediator on that causal pathway. And so we performed our epigenome-wide association study where we just regress methylation levels on smoking status. We control for various confounders. We also take some extra steps to deal with potential bias for residual effects. And then we apply very strict multiple testing corrections. In this case, we use the Bonferroni cutoff to identify significant sites. And this is called the volcano plot. So on the x-axis is the effect size. So anything to the left of 0 means you have lower methylation associated with exposure to smoke. Anything to the right of the 0 means you have higher methylation associated with exposure to smoke. And then on the y-axis is the negative log 10 p-values. So the dots that are at the very high end there are having very small p-values. And since it's negative log 10, that 40 means that you have 0 point and then 40 zeros before you get to that first number. And so our most significant hits were actually at minus 119, minus 42. So essentially the strongest p-values I've ever seen in an environmental study, aside from other studies of smoking. So one, very exciting. I think these are very statistically robust findings. But the other thing I want to highlight is we see this massive difference in methylation at this top hit as well, where it's about 20% lower among those that were exposed to smoke. The way you can kind of think of that is that's like 1/5 of the cells in that sample showed a different methylation state in association with smoke exposure. But overall, we see a lot of different hits showing up here. It's not just these couple of really significant ones. And one of the questions is, there's lots of CPG sites within individual genes. So is this just telling us about a few genes where they have a consistent change in methylation, or is it happening throughout the genome? And so we use these Manhattan plots to identify what's the genomic organization of these findings. And we actually see that there's actually quite a few individual CPGs throughout the genome at different chromosomes and in different genes that are being responsive to this exposure. And so it's not like we're just seeing a few genes with a consistent epigenetic response, but instead we're seeing lots of different regions of the genome potentially affected by this exposure. So what we want to understand next is whether or not this is just a signature. Is this just like a fingerprint of smoke on the chemical, the epigenetic regulation of the genome, or is it actually having a biological effect? So we performed these enrichment analyses where we're trying to say, among the CPGs that we detect, do we see more or less than we would expect of certain regulatory elements? And so first we tested for enrichment of these partially methylated domains. And the way to think of this test is this gray bar here is the proportion of CPGs on the entire array that are annotated to a partially methylated domain versus the proportion of CPGs in our list that was identified in association with smoke exposure that are annotated to those. So if the colored bars are smaller than the gray bars, that means it's depleted, meaning there's many fewer PMD CPGs in our hits compared to the overall array. And that is what we see here, that there's quite a few less, there's a much lower proportion of PMD annotated CPGs among our hits compared to the total array. So that might be an indicator of less of these placenta-specific repressed regions showing up. And so then we tested for other genetic regulatory elements as well. So it's a little harder to read. This is a series of different regulatory elements, but where we see the most dramatic effects is when we focus on enhancers. So 06 and 07 here. Here, the gray bars are very small, meaning there's not a ton of CPGs on the array annotated to these different enhancer regions. But among our CPGs that are associated with smoke exposure, there are a lot of them that are annotated to enhancers. And so the overall takeaway from that is that the CPGs that we identified are in these very active areas of the genome. And so we don't think it's purely an environmental fingerprint occurring in the methylome, but this is likely related to some biological responses to the exposure as well. And so last, I'll do a real quick review on this one, but we then tested that in one of our cohorts where we had paired RNA-seq data as well as methylation. And this is where we're just looking at whether or not the CPGs and their methylation state is correlated with the gene expression value for genes that are nearby. And so again, negative log 10 p-value on the Y. On the X is the distance between the CPG site itself and the transcription start site of that gene. And so what we kind of see is that where we see these relationships between expression and methylation are really occurring at these CPGs close to the promoters of those genes. And we see the kind of hallmark interpretation of methylation expression, which is that those CPGs are often, so the higher values of methylation are associated with repressed expression. And that's represented by these red dots, and only a few where we see that inverse relationship. And so overall, our interpretation here is that we identify a methylation signature of smoke exposure during pregnancy, and that signature is likely representative of biological activity. And our top hit, which had the 20% loss of methylation associated with smoking, is within this LEKR1 gene, which is inversely associated with methylation. And so these are some interesting findings. Unfortunately, this gene is not very well studied, so there's not a lot of information for us to refer back to understand if it's biologically related to what we would expect to see for smoke exposure. So I think this is some interesting potential future directions for experimental studies to characterize, well, what happens if we repress or overexpress this gene, and would that make sense in terms of responding to environmental tobacco smoke, or as it relates to growth regulation during pregnancy. And so last, very briefly, on the y-axis here, we wanted to compare, is this signature we identify completely unique to the placenta, or is it similar to what others have shown in cord blood? And so these are comparing the coefficients between our study on the y-axis and another study of cord blood responses to smoke on the x. And if you saw a similar pattern of exposure, you'd expect to see a nice kind of positive linear trend here, and we don't really see that. So on average, we see a very unique signature in the placenta. And then despite having 443 identified significant hits, only 6 of those are actually showing the same direction of effect in that cord blood study, and also having a high or a small p-value. And so we have this very tissue-specific finding. One of the only genes that showed up within our overlap of these 6 CPGs was the AHRR gene. And this gene is involved in the aryl hydrocarbon receptor pathway. That pathway is primarily involved in detoxification. And so it actually makes sense that maybe a detoxification pathway would show a similar response independent of tissue, whereas maybe other systems that aren't specific to detoxification would be very unique between tissues. And last, we then screened whether or not our 443 CPGs were also associated with various birth outcomes. We find that 1/3 of them are, and most of them are associated with lower gestational age at birth. But there are quite a few also associated with birth weight and length as well. And so in parallel with some of the authors on that initial project, we were performing this mediation analysis. And in this study, we identified that the impact of maternal smoking on low birth weight was mediated by variation in DNA methylation at 7 different CPGs, and it could explain maybe around 30% of that relationship. Now, there are caveats to this. I don't necessarily know that what we're seeing is a causal mediator, but it is at least representing a statistical relationship between tobacco smoke and lower birth weight, and it's in this biologically plausible tissue of effect of placental epigenetic regulation. So the main conclusions from this study was that the placental methylome is very responsive to maternal smoking during pregnancy, that this is occurring in these very active regions of the genome, that these could be intermediate markers between maternal smoking and birth outcomes. In our study, we've really only looked at low birth weight, but I think based on what we see in the output from the meta-analysis, I would actually like to investigate this more with impacts on gestational or at birth timing, because that's actually where we see the biggest signature now. Almost 1/4 of the CPGs that were associated with smoking are also associated with shorter gestation. And so overall, the main point of this is highlighting that epigenomics and focusing on the placenta can be this powerful pairing for studying the impact of prenatal exposures. And this is because the placenta exhibits developmental plasticity. It's adaptive to the maternal and the fetal signals, as well as environmental exposures, and that proper placental function is critical to achieving a successful pregnancy, as well as appropriate fetal growth. Now, I'll spend a few minutes highlighting some of the ongoing work in this field, and I'm going to talk about this Victor project that Kevin mentioned earlier. For this study, we're trying to study the growth and metabolic programming of prenatal PFAS exposure by looking at the role of the placental epigenome and potential protection by maternal exercise. And so I'm kind of leading aim 2 of this project, which is trying to understand what's the placental epigenetic response to PFAS and what are the impacts on gene expression. And so as a very brief background, PFAS are something that I think a lot of people have at least heard about in the news. They may not be very familiar with them, but they're these long chain chemical structures with carbon chains and then fluorine attached to them. And this structure provides them with a unique property of being able to repel water and grease. They're used in a lot of industrial and everyday products like upholstery, cleaning supplies, but they're also used in firefighting, and they're very prevalent in use for at Air Force bases and military bases as well. And PFAS have been shown to be associated with lower birth weight as well as shorter gestation. And their structure is actually somewhat similar to some like estrogens and progesterones, and they're thought to maybe have some endocrine disrupting effects because of this. And because of that, they can interfere with different hormone systems. And as we know, the placenta is a very active endocrine organ. And so what we're doing right now is we're actually having this flood or this avalanche of data that we're generating for this project right now. So in 6 or 7 months, I will have a lot more to talk about, but I'm just going to highlight a couple of the ways that we're thinking about our analysis here. And we can perform something like an epigenome-wide association study like we did for the smoking project, but we're also going to take some approaches here like a gene network analysis, which can take our 13,000 genes that we've detected and then cluster them into about 20 different modules that are representing genes that have similar expression patterns. And those are thought to represent biological activity or genes that may be the target of specific transcription factors or regulated by microRNA. And so right now I have a student that's studying how these networks look within the placental tissue. And then once we have all of our PFAS data cleaned, we're going to test whether or not the placental PFAS are associated with these different co-expression patterns. We can also use our epigenetic data that we've measured to estimate placental cellular composition. And so cellular composition of the placenta changes quite a bit throughout pregnancy, where over time, certain cells called the trophoblasts fuse their cell walls and become something called this large syncytiotrophoblasts. And so this is a unique property of the placenta that just changes with gestational age. And so if PFAS is actually having an effect on gestational timing, we might see something within these estimated cellular compositions as well. So we're going to do some exploration of whether or not PFAS are associated with changes in cellular composition of the placenta estimated from methylation. And then a rapidly advancing area is in epigenetics in general is related to biological aging, where you can develop these algorithms. And once you know a person's methylome, you can actually predict their age quite well. And this has been done with placental tissue and gestational age. And so in our data, we've now estimated the sort of epigenetic age of these placentas, compared them to the reported ages here. So these are 3 different clocks that are trying to estimate that age. And the deviation between the reported and predicted age is thought to represent something about biological aging, or in the placenta, the case of the placenta, we think it might be related to developmental states. And so I have a master's student that has just finished her thesis looking at this. I'm saying it's still fairly preliminary, so I'm not presenting those results, but it does look like this deviation between predicted and reported age is related to birth weight and postnatal growth. And then the next steps will be to see whether or not it's also associated with PFAS exposure. And I definitely want to highlight all of the great contributions. My work certainly wouldn't be possible without my great mentor from my postdoc, Carmen Marcet, a lot of the collaborators that helped make that meta-analysis possible. And then, of course, our Victor consortium, Alina Andres, who coordinates the cohorts that we're working with, Kevin, who is actually a much better writer than he led on in the introduction, and then Kartik, who has developed a lot of our placental resources for this project, and then Andrew Morris and Dana Barr, who are measuring the placenta or the PFAS levels in this study. And then, of course, my lab, who's been helping out with this work, lab manager, data manager, and then students that are helping me sift through all the data that we're getting right now. And I definitely have some time for questions. So.
Oh, did I unmute? Okay, it wasn't unmuting for me. Todd, that was awesome. I can imagine that you're an excellent lecturer. I would think that your students at 1 o'clock are in for a real treat, but thank you. So I know there'll be other questions, but I guess I had one question about whether or not within any of those cohorts that you have, or any cohorts that exist, if anyone has, term pregnancy as a inclusion criteria, I guess, right? Because when we did our earlier studies with smoking, we actually only recruited in individuals that were full term. And we still saw reductions in birth weight and reductions in birth length, for example. So are there cohorts that you could look at to follow that, I guess, hypothesis up without having to just do it statistically?
Yeah, so in that study, the vast majority of the data are term pregnancies. I think all but 3 cohorts actually excluded preterm. So we did the analysis first overall with without excluding, and then a secondary analysis. If we exclude preterm births, we see almost the identical relationship. So it's not being driven by preterm birth.
Okay, Ellen, if you just want to go ahead and make a comment or a question, go for it.
Yeah.
If anybody else wants to open their camera, then I'd know who has questions. If anybody has them, or raise your hand, please.
Yeah, Todd, that was really interesting. I had to step away when you were talking about the method, so please excuse my question. But are you looking, so how are you operationalizing maternal smoking or exposure? I think you talked about it as tobacco smoke exposure. How in your study is that operationalized?
Yeah, so in that study, it's self-reported during any of the 3 trimesters as the main exposure. And then the secondary analysis is excluding those that only reported smoking in the first trimester, and then only looking at those that reported smoking throughout the entirety of their pregnancy. So there's no question that we're susceptible to misclassification here.
Yeah. Yeah.
But I
Well,
think
tobacco smoke, I mean, so it's really firsthand smoking as a
yeah.
proxy. You really, so one of the limitations, or one of the things I encourage you to think about, is how to capture the secondhand smoke exposure. That's always all of that tobacco smoke from an environmental health perspective. You know, so just because they're non-smokers doesn't mean they're not exposed to tobacco smoke, either at work or at home, or so.
Yeah.
Yeah,
I
and
think
I know John's popped on here. He's looked at that, I think, as well. So
yeah, quick comment on that.
And then,
Totally
yeah,
go ahead, Todd. I'm sorry.
I'd say, yeah, I agree. This is one of the issues of trying to do a large scale meta-analysis as well, is we're dependent on the data that have been collected rather than choosing the best data to work with. I will say there's been secondary studies looking at NNAL, and when we've seen that, we see kind of the same signature. There's some variation in what pops up, but at the same time, that's lower powered for a much smaller sample. So I think kind of collating across these studies would give us the best picture in the end.
Hey, Todd, nice to meet you. Great work, man. And that's a lot of ground to cover. I have some generic questions for methylomics folks like you. The first I wanted to make a mention regarding what Ellen mentioned. We did RNA-seq on leukocyte DNA from kids exposed to secondhand smoke, and it's bonkers. Like, it has monstrous impact. Like, if you look at a heat map from the basically the dot plot kind of format, it's just incredible how broad-based secondhand smoke can create alterations in kid leukocyte DNA. And quite honestly, I like going beyond that and trying to decipher things gets pretty much like a swamp for us, to be honest. But let me ask you this: one of the key things for these omics efforts is the cellularity of your method and your sample. So, for example, cord blood is a great example where you could have a 5-fold deflection in the neutrophil to macrophage prevalence. And so really you're looking at cell averaged responses when we know that that's probably not accurate, since T cells, neutrophils, and macrophages have fundamentally different algorithms. So in light of that reality, have the methylation community members like yourself moved towards cell sorting before blasting through these things, or just trying to clean up the cellularity variance hiding in your data, which makes, in my opinion, very difficult interpretations?
Yeah, great questions. And so I'll say there is some movement in that area. It's actually particularly hard to do for the placenta. to get the collection in a way that you can then do the cell sorting and retain the signature. And so one it's definitely difficult. I have a collaborator working on that right now. I'm not involved in that project. So there is some movement there. The way we deal with it here is there are cellular references of the methylation pattern, so we can estimate what the composition of our tissues are. They're not great because they can only pick up the cells that we've already well characterized. And so in the placenta, that's like 7, I think, that we can estimate. There's over 40 different cell types in the placenta. So it's a rough estimate. What I would say is there's some movement there. I don't think we're at the point where we can do it perfectly, or actually some labs can do it well, but not very many right now. What we can learn from this type of work is going to tell us that averaged effect, and where we see something as strong as smoking, I think it still works well, because even if it's only occurring in one cell type where we're seeing that effect, it's going to come out in the average anyways, as long as we have a large sample size. So I think what we're learning is still robust, but we can't take it back to know which cell type is having the effect. There are some statistical methods that give us an indication there. We're exploring those. I'm not sure how well they work right now. So we're kind of keeping it at the average level until we have, I think, advanced in that field a little further.
I think anyone in this audience that's thinking about going in this space, that's going to be the next riddle, man. I think that drilling down on cellular algorithms of these complicated omics platforms is absolutely going to be a huge opportunity. You're in great position to do that work, Todd. That's terrific stuff.
Yeah, thanks.
Yeah, not even with just the cellularity, I guess just even like with the blood flow and like where those cells are located with the vessels and things could be really telling, I think, too. But so Todd, I mean, I guess the placental age stuff, I mean, we've talked about within our Victor quite a bit. And I mean, I think that's extremely exciting. And I guess, is there a way, I mean, I know you said you have it within the cohort, but is there a way to predict whether someone actually quit smoking, even if they said they did, right? Like, can you come up with like a predictive model of like, all right, this person said they quit smoking, but like they look exactly like someone that smoked until the day this baby was born? Or is that just like too fishing that you can't really do something like that?
Well, what I would say is right now, I don't think we can do that now, especially now with the placental tissue. There's not been enough data to develop that type of predictor. But I love that question, because we actually, in one of my other studies, looking at former smoking before undergoing IVF treatment in the follicular fluid, we're measuring the epigenome, because they can't undergo IVF if they're still smoking. Now, among them, we can detect a signature of former smoking. And at some of these CPGs, you see this really striking correlation between the time since they quit and the dose that they were smoking at, even once they've quit. So there are some of these possible indicators that will give us that information. It's been studied in blood and current and former smokers as well. I think the maternal smoking side and placenta, it's not as advanced there yet, but I think there's opportunities to develop those tools, which in Victor, it'd be great if we could, but it's all non-smokers, which is
well,
for many reasons.
I see Ellen unmuted, but I just wanted to say that you should probably, I didn't see Pat Hannon on here today. I think he's trying to put in a supplement, but he would be someone probably that you would have some interest in as well. He's doing exposures in IVF and looking at fertilization and those types of things. So he would probably be a great collaborator potentially in the future too. Ellen, did you have another comment? We have about 2 minutes because then Todd's got to hop off, but I did want to say that Todd is going up for promotion and tenure at Emory. That was one of the reasons I wanted to make sure he got some national invitations. So if anybody does get a letter request for writing a promotion letter for Todd, it would be great if you accepted that. I don't think I'll be able to write one because we collaborate, but go ahead, Ellen. Thanks.
That's funny. Jason's asking my question. Great minds, Jason. Can you measure nicotine or cotinine in placental tissue?
I don't know. That's a really good question. I would have to ask my
yeah.
I would ask my so Dana Barr is our chemist, our organic chemist. She I would ask her, she would know.
Yeah, because that could get kind of at Kevin's issue, because you could look at the interval level data, right? Not just smoke or not smoke. I assume that's, and that's not very powerful way to ask it. So you could look at exposure, over nicotine, you could look at the interval interval level nicotine or cotinine level.
Yeah. Yeah, I think that's a great idea. I'll see if that's an option for us. I'm not sure.
All right, awesome. Well, I know Todd has the lecture at 1, so we'll close it down here. But thanks everyone for coming. I know we have a very busy week with the Wyatt Symposium tomorrow, and we had some other symposia this week as well with the Unite and others. So thanks for coming, and thanks a lot, Todd. Look forward to seeing you down in Atlanta next month for our retreat.